I would like to welcome you back. What we are going to be talking about in this section is other types of experiments. Because remember, as I mentioned before, there are times where we cannot use this classic method of investigation. And most of the research we've been talking about has been describing kind of this classic approach, or this ideal approach where we control all of the control variables and then just change one of the independent variables. We would also have these large sample sizes, and our experiments would work every time. But in the field, especially in biology, this is all but impossible. For example, in ecological research, we cannot control all the independent variables of an entire ecosystem and manipulate only the one thing we want to test. It's just impossible. So how do we go about conducting research with this limitation in mind? Well, we use other types of experiments, such as natural experiments, field studies, modeling, and a variety of other techniques. And each one of these have positive aspects, and each one of them have their own negative aspects. For example, if we're interested in how a volcano will affect the local vegetation or even the economy of an area, we cannot make a volcano erupt and then sit back and go, oh, look, those people are running and dying and burning, and oh, look, it's burning up all the plants, and oh, yes, the economy is going to be devastated by this. No, we can't do anything like that. That'd be awful. I mean, I guess we can make a volcano erupt if we wanted to, but, we just wouldn't do that to see how it's going to affect an entire economy of an island or, a part of the country, okay, or even just the vegetation. But if that's what we're interested in looking at, and the volcano does erupt, we can then observe the effects of, what it's doing to the vegetation, what it did to the soil, how long does it take for the soil to recover, how long does it take for people to move in, how long does it take for that economy to recover, all of these things we can then observe to see what is actually happening. Now, in field experiments, we might go out into the field and look at plants or animals and whatever we're interested in and try to manipulate those animals or their habitat. For instance, if we're interested in the effects of removing all of the trees in an area, what is that going to do to the deer, the white-tailed deer in the area? We could go in and choose similar areas and remove the trees from some, and then leave some areas where we don't remove the trees. Remember, we want these control areas or these control groups. We can then measure the deer's response. Do the deer increase their reproduction in the areas where we remove the trees? Does their reproduction decrease? Do they just leave? Do they starve? Whatever's going to happen, we can then observe it. But remember, we can't control everything. But when we set up our experiments, we have to control as many of the variables as we possibly can. And we also want to have some sort of control plots where we do not manipulate. Okay, so we want, our experimental group where something is going to happen to them or their environment, and then we have a control group where nothing is being manipulated, only measured. And controlling variables is fairly difficult in these natural experiments or these experiments that are conducted out in the field. We can cut down the same number of trees in each area, but we cannot control everything else. Okay? Are there turkeys in the area that will compete with the deer for food, and maybe they're not in the control area? Do people visit the area? Are all the plants the same? Or if we cut down the trees, in one area we might have invasive species grow up, and in the other area we have all sorts of natural species grow up. And this is something that we can't necessarily control. If you look at any two places in the natural world, they are not going to be identical. So these differences cannot be easily controlled for and can cause problems when you try to interpret the results. To account for this, we try to replicate our studies and have multiple controls and treatment plots where we cut down our trees. And while this study might not be as controlled or as precise as our laboratory experiment, a natural experiment might ultimately prove more realistic because the results occur where the animal actually lives. But to be effective, what we've got to have are these multiple replicates where we replicate what we're doing over many different plots, or as many as we possibly can do. Now, if there are no experiments that you can figure out to do in the field, you can use mathematical or computer models to try to figure out what would happen if you could conduct an experiment. So let's say that you wanted to know the effect of cutting down trees on the endangered red-cockaded woodpecker. They are not, the U.S. Fish and Wildlife Service are not going to let you go in and destroy habitat for these endangered birds. But if we, in general, know what kind of response we think they would have, or we can manipulate another similar woodpecker that's not endangered and use some of that data, then maybe we can develop some sort of a model for what would happen for these other birds. Now, so you're going to need some sort of information about what you're studying. Let's go back to our deer experiment again. Let's say that we were not allowed to cut the trees down just for our experiment. We could use a computer model to look at what would happen to the forest and what would happen to the deer if we did. But for this to be accurate, we would have to know a great deal about deer eating habits, preferred food, how the vegetation would respond to the trees being cut down, what animals live there, and all sorts of other information that could go into our model. Because the better the information that we have, the more accurate our model is going to be. Now, our knowledge and ability to conduct experiments is also imperfect, and we have to make do with the research methods that are available to us as scientists. Luckily, as time passes, technology increases, and we develop new tools that we might be able to use to conduct additional experiments or get further insights into the systems that we are studying. That is why I keep referring over and over again to science as being a process. It's a scientific process. It's not just a one-off thing that we do where we conduct one experiment and we know the answers to the questions that we have posed. You know, especially when we're looking at something as complex as biology and as difficult an area to do research in as biology. So, you know, you've collected this information, you've did your statistics, you've written your paper. How does this information actually get out to the world? The primary sources that are used to get science from the scientists to the world are peer-reviewed journals. And this is what I mentioned before. This is information that's being judged by others to have at least met a particular standard. Most of our information, though, comes from secondary sources. These are media shows, newspapers, blogs, web pages, and other information outlets that somebody hopefully has read the primary literature and written an article about that bit of research. Now, something that's important to remember is that the people who write those stories may not fully understand the research and may misinterpret it. If you get your science information from these sources, you have to be aware it may not be 100% accurate. And, you know, this may not be purposeful. They may have not have done this on purpose, but the information may not be accurate or may be more complex than what they reported. For example, if their media reports that chewing a particular kind of bark will reduce the common cold by 50%, we would think, man, that's freaking awesome. Where do I buy this miracle bark at? If they fail to mention that this was a study on an indigenous South American tribe who had been previously determined to have a more robust immune system than most normal North Americans, then this bark may have not been the primary cause for the reduction in the duration of their colds. You may also need this souped-up immune system as well. The point is, don't believe every science report that you hear. If you want to be really sure about what the report is saying, you need to track it down to the primary literature and read and understand that. Now, just to conclude this section on scientific information, this module, all it's designed to do is give you a brief overview of how scientists develop and collect information. If you become more interested in the sciences or becoming a scientist, you've got to spend a significant amount of time in college learning how to set up studies, how to conduct statistical analysis, or at least, know where to find these folks who can do this. And it may mean learning to work with particular animals and spend lots of time in the field, or learning how to set up laboratory experiments, or running DNA analysis, and all these different things. There's just this phenomenal amount of information that goes in even to setting up and performing a relatively simple experiment. But hopefully, what this module will do was just to get you thinking about how the information that we're going to use in the rest of this class and in other science classes, how that information is actually developed. And it's not just something that, somebody who calls himself a scientist just looked at and said, oh, well, this is the case. It's actually determined through a process over time and with many people and the input of many people. So I hope you found it interesting or at least educational, and that'll pretty much wrap it up for how we develop science or scientific information.